We are ready to begin our second session. To chair the second session, I would like to invite Dr. Gianluca Bona. He studied physics at ETH Zurich, where he received a PhD degree for his investigation on surface magnetic structures. Subsequently, he joined IBM Zurich Research Lab and did research in intense high-speed semiconductor lasers. In 1994, he did work on integrated optical devices, which led to a series of reconfigurable planar light wave circuits and photon band wave concepts. Since 2004, he is Department Group Manager of Science and Technology at IBM's Almaden Research Center in San Jose, California. I would like to welcome Dr. Gianluca Bona.
Thank you, Dharmendra. It's a great pleasure and a great honor to be here because, as you heard, I'm a physicist that still believes in the good tools that physics and mathematics have developed over the centuries. But I'm also very aware that our tools are limited and we have to reach beyond that and integrate much more from that small world of physics. And that's why I was so excited when we got the proposal to have these discussions and workshop in 2 days about cognitive computing. And I'm really pleased and honored to introduce now a person that has worked at the interface between physics, used physical tools and computational tools to bring the field of neuroscience further. It's Professor Dr. Henrik Markram. He's co-director of the Brain Mind Institute. He's director of the Blue Brain project at EPFL, École Polytechnique Fédérale de Lausanne. He is director of the Center of Neuroscience Technology. Henry Markram has been, before joining EPFL, in various research roles in the Weizmanns Institute. He was an early tenure a Stanley Helen Diller Professor of Neuroscience. He was also working for a couple of years as the Minerva Fellow in the laboratory of Nobelist Bert Sackmann at the Max Planck Institute. Among his many contributions to the fields, I just want to mention one. It's the precise millisecond relative timing of single pre- and post-synapses action potential to reveal a highly precise learning mechanism operating between neurons. This is what people in the field call today as STDP, spike timing dependent synaptic plasticity. So these many fields that Henry Markram have worked in has also led to a series of prestigious awards such as the Ebner Science Award, the James Heinemann Prize, the Abraham Research Award, and of course he's now leading the Blue Brain project that is connecting EPFL and IBM in this field of neuroscience. And it's for me a great pleasure to introduce Henry Markram, who will talk to us about the emergence of intelligent in the neurocortical microcircuit. Please welcome Henry.
Thank you very much for the introduction, and thank you very much, Damudra, for this honor of this invitation. So, about 3 weeks ago, we actually had a conference in Barcelona, which was to commemorate the 100 years since Ramon y Cajal started drawing every neuron in the brain. And he essentially did. He drew every neuron, every type of neuron that we basically know now. The purpose of this conference was actually to reflect back on the past 100 years, but also to look forward into the next 100 years. If 100 years can happen, well, then you can look a little bit further and try and think what's going to happen in the next 100 years. Well, two very interesting issues or topics of debate arose in this meeting. And the first one was, many people said, what would we do as mankind to put inside a time capsule and send it out to space so that it would land on some alien planet and they would be able to understand who we are? So, of course, you had all kinds of opinions. You had people saying, let's send the genome. We send it all there. So you're going to read the genome. I don't know. We don't know how to read the genome. We don't know how to reconstruct who we are from the genome. Of course, it's a very important milestone, and it's an important thing to send. But are they going to understand who we are? Other people said, because it was, of course, a meeting about the cerebral cortex, about the brain, all the different types of neurons, they said, let's send all the piles of books that show the different neurons of the brain. Okay, you could send that. We can't even look at all these thousands of papers and reconstruct the brain and work out how it works. So do we think that they're going to be able to do it? Maybe if they're very advanced and they've already seen that kind of biological design, they may say, yeah, okay, this is easy. But maybe it's a silicon race, and they can't work out how we're built. So, of course, there were also other people, and they said, let's send DARPA's robot. So if you send DARPA's robot, are you going to put your name on there and say, this is us? So, yes, they would recognize that we're a very advanced race. We can build very sophisticated robots. We could send Darwin X, and they would know that we can build very sophisticated robots. But is that really going to tell them about us? The bottom line is that what we think is that the one thing that would really demonstrate to an outsider that we really understand what the brain is doing is if we sent them the blueprints to build us. I don't mean the biological blueprints, but that we've actually understood all the biological processes to build a robot that will look like us, interact, and have all the cognitive capabilities. Of course, that's a dream, it's a challenge, and that requires to build a robot with a brain that is virtually a copy of the way that we are. And so the first part of my talk is going to try to illustrate to you the very first small step towards not just building a neural circuit that is very clever, but to building an accurate copy of a small part of the brain. The second issue, which was a very interesting issue that was raised, was what makes humans special? So you had all kinds of reactions. Some people said, humans are not special. They're not special because in each environment of a different animal, we all adapt as well as we can within that environment. But the overriding opinion was that we are special. We're special because we can share knowledge for the purpose of understanding our own brain. And it's difficult to find that animals could do that. But I think that there's another reason why we're far more special than that, and that is that imagination is imagination. Imagination is more important than knowledge, as Einstein said. And I think the reason why imagination is so important is because it allows us to look inside the paradigm within which we are investigating ourselves. And not only to look inside that paradigm and say, this is the foundations, the axioms that we stand on, that we use to interpret the brain. But it allows us to step outside of that paradigm and saying, what if we actually interpreted the brain from a completely different place? So in the second part of my lecture, I'm going to try to take you and illustrate briefly the current paradigm that rules, the paradigm from which we interpret how we understand the brain. And then take you to the opposite paradigm. So let's begin with the first, as we heard beautifully from Jerry Edelman, that the human brain really is because of the neocortex. The human capabilities because of the neocortex. That is the emergence of man from mouse to man, and it's a 1,000-fold expansion in the neocortex. So much so that 80% of our brain now is neocortex. This is what defines, this is what allows us to sit here, to interact, to predict, to analyze. It's the neocortex. In the neocortex, here. The neocortex is essentially composed of small little microchips, or columns, which are known as columns, functional columns. There are about 1 million of these columns in the human brain. Each of these columns is about 0.5 millimeter in diameter. And one can debate forever about what is a column. But the bottom line is a column is the minimum set of neurons that create a society for neurons, because neurons are not islands. They need a group of neurons around them. And the minimum set of group of neurons around them turns out to be approximately the size of a column. In the rat, it's about 10,000 neurons. In humans, it's about 100,000 neurons. In evolution, these columns were added until we ran out of space and had to keep folding in on the brain. And that is one of the reasons why the brain is so highly convoluted. We just kept adding these chips. So the secret to the neocortex is to understand these little columns. And the goal of my lab in the past 10, 12 years has been to go inside these little columns and try and reverse engineer how are they built with exquisite detail, and as much detail as we had technology that would allow us to do. So just to give you an idea what that looks like, this is a technique that we developed at that time in Bert Sackmann's lab. It's called multi-neuron patch clamp. You put a slice of the brain, you put it under a microscope, you use infrared optics to visualize the brain while it's alive, this piece of the brain. You use a glass pipette to approach the neuron, clearing the way by positive pressure, and then you patch on, you suck onto the tip of the membrane, and you then blow a little hole in the membrane, and then you have electrical continuity. You can record the electrical behavior. You can inject a dye into it so you can see the physiology, the morphology, and you can record from many of these cells and start sending little messages between them. This is an extremely powerful technique because for the first time it gave us the tools that we can systematically start reverse engineering how the microcircuit is built. So over the years, we've developed this to the most extreme art that is possible right now with the technology. And this is a 12-patch setup. We can't get more manipulators in there. We need to have very fine sub-micron precision of control of these manipulators so that we can choose these cells. But this has allowed us to rapidly increase in the rate at which we get the data to work out how the circuit is built. This is just what it looks like if you now inject a dye into these and you use a confocal laser scanning microscope, and you can see what is the full structure of these cells. So the first thing that you hit upon is you rediscover Cajal's inspiration that he must have had when he looked under the microscope. And that is that, and he called them the butterflies of the cerebral cortex, is that you have a whole spectrum of different types of cells. It's the same, you go into the Amazon forest, you have a whole spectrum of trees. And here you do the same. This is a daunting variety of trees, and when you go to the computational people and you show them this is what it is, they, of course, are completely flabbergasted. How are we going to understand the brain with all these different types of neurons? Well, it turns out that they do cluster and group into certain classes, and basically there are 9 types of what we heard in the previous lecture as inhibitory cells, and there are also about 9 or 10 types of excitatory cells located in different layers. So there is an order, and it is not that complex. It's just that each of these types are different. And of the 100 billion or 30 or 100 billion neurons in the brain, there's no neuron that is the same. It's like people. There's no person that is identical, even for identical twins. No neuron is the same, but they do fall within classes. So we can now model these cells, any one of these cells. And this is an example. What we have to do is very often we need to repair first. Before we model them, we have to repair them because in the slice, we may shave off some of their branches. And this is just showing, for example, an algorithm that we use to repair the dendrites, which are in red, where neurons receive, and the axons. Once you've studied 100 pine trees or 1,000 pine trees, you don't need to get every 100 billion one of them in the brain. You can clone them. So we can essentially now clone as many neurons as we need of each of the types with the full morphological diversity that we see in the neocortical column. So we have the neurons. The next thing is we need to be able to capture the electrical behavior. We inject when you inject positive current into these cells, this is a positive step current. The cell, of course, depolarizes. It's called depolarizing because all cells, including plant cells, sit at about -70, -80 millivolts resting potential, and then suddenly they spike. And these are the action potentials that is thought to be the currency of the brain, thought to be the basis of perceptions and consciousness. Neurons produce action potentials. But if you look at the neocortex or any other part of the brain, you discover another big challenge, and that is that they all have different behaviors. Not only do they look differently, but they also have different behaviors, and we need to now understand these different behaviors. Some of them have stuttering behavior, some of them wait before they start firing, and some of them have bursting behavior, and so on. We've characterized most of these different behaviors, like a psychologist, I suppose, characterizes all the different patients. And what we've seen is that each of the anatomical types can also have multiple electrical types. So you may have a certain anatomical, a pine tree, but that pine tree may have different behaviors. And if you put that together, you start to see that in the neocortex, in one layer, you have many different types of neurons. These are the names of all the different types of neurons that you find in one layer. It's about 50 types. So you're looking at about over 300 types of neurons in the neocortical microcircuit. We can capture them, each one of them again. They may fall within a class, but they all are slightly different. So not only they're structurally different, but they're also electrically different. So now our challenge was we need to be able to model not only the classes, but all the different variations within a class. So what we had to do is to start going further below the surface and find out exactly why they're different. Now we know that the neurons are different because they have different behaviors, because they use ion channels. They have ion channels for sodium, calcium, chloride, potassium. And there's in the neocortex, there's about 200 ion channels that they can select from. And if I select 20 of these, I'm going to have a certain behavior. And if I select 20 of those ion channels, I'm going to have a different behavior. So we have to work out now the 200 ion channels. We have to be able to model each one of them. And then we have to know which one corresponds to the different behavior so we can build each of these neurons. So over the past 10 years, one of the techniques that we've used and developed was to go into these cells and extract the cytoplasm, the juice from inside. And it's like a reverse copy process. We take the messenger RNA to reverse copy a complementary DNA of the genes that are switched on in that single cell. So we can use primers to find out which genes are switched on in that cell. These are names of ion channels that are used, and we can see that these genes are switched on to produce those ion channels in that one cell. Now it gets more complicated because these genes produce subunits, and these subunits can combine in a combinatorial manner to give you over 200 ion channels. So we've had to work out how they combine, what types of ion channel combinations are used in the neocortex, and we've mostly done that and been able to, we start now to build a large database of these different types of ion channels, and we know now which ones of these we need to use to model each of these different types of behaviors. But it's not that simple. It gets a little bit more complicated, and that is that we need to, neurons are very complex structures in terms of their trees. And these ion channels are inserted in different locations. And depending on the locations, this can add new functions to the neurons. So we not only need to know which of the 20 or 30 ion channels they're going to select from the pool, we have to know exactly where to put these ion channels. So for a whole neuron, you're talking about several million ion channels, several types, and you have to know where to put them. So we use many, many different techniques, and I won't go into all of them, but this is freeze fracture scanning microscopy, and you can actually locate single channels, single molecules, to constrain the model and say that there are certain distributions and certain locations for these ion channels. So we think that, and we're in the process, that it will be possible to start putting these ion channels in exact locations. We've recorded from over 10,000 cells, and we've begun to start deriving what we call the first recipe of the neocortical column. The recipe is the recipe of cell types that are used to build up the circuit. Now, as I said, the neocortex from mouse to man, this column is surprisingly stereotypical. There are many, many features, that approximate ratios of excitatory to inhibitory types of cells, the types of interneurons that there are. But the subtle recipe may change across species and across regions, but essentially the core aspect of this recipe stays very much the same to give you a column. So once we have this template, we would be able to begin building columns from different species, different brain regions, and so on. The next thing that we need to be able to do is, of course, this is an infrared picture of the whole column, and this is what it looks like if you just pack in a few of these cells. Red are the dendrites, blue are the axons, but we now have the neurons. We can model their structure, we can model their function, but how are we going to connect them? So we've done a number of different types of experiments, and the first is that what we wanted to know, Jerry Edelman raised an important issue in the neocortex with so many specific things. Is it possible for one neuron to go and target a particular cell? And he's in the past made a very clear claim that it's not possible. So we wanted to check this experimentally, and what we did was to record from two cells, fill them with a dye, and use a confocal microscope to do a very, very detailed three-dimensional reconstruction. And then we take the axon and we track it, we follow it, and we count every time it gets within 0.1 micrometers of the dendrite of a target cell. And then we do statistics to see whether for connected and unconnected cells there's any bias as to how the branches are going to target their different cells. And as Jerry would have predicted, there is no bias. Every axon touches every target cell. It's an all-to-all circuit that is ready and in place for a specific functional circuit. And we could see that because it's not going to transmit information to all of them. It's just structurally in position. It's only going to transfer the information to about 10% of them. And those 10%, all it has to do is grow a synapse. And I'll come back to that at the end and show you that that's a very dynamic process. Now, we know we use that, and we're going to come back. We're going to use that information about all-to-all structural connectivity with certain numbers of touches on each cell to constrain the way we position these neurons in 3D space when we build the circuit. But we need to know more than that. We need to know for each type of cell not only that it touches it, but exactly where it touches it. What part of the axon is used, and on what part of the dendrites does it place those synapses. So what we've done is to record from lots of cells that are connected. This is a small basket cell that is connecting 3 pyramidal cells, and the red shows points of information exchange, synapses. It looks like a mess, but if you do this repetitively for the same type of pre- and postsynaptic neuron between a pine tree and an oak tree, you find that there are patterns. They're very specific patterns in terms of which parts of the roots are used to contact which parts of the branches. And this information is the most crucial information to building a biologically accurate circuit. Nobody in the past has bothered to collect this data because it's extremely tedious. But these are the locks and keys that will tell us and allow us to place 10 to 20 million synapses within the size of a pinhead in exactly the right location, so that they will exchange information in the right places. And we're going to use that. So what we then did was to build a circuit builder, and it's an application which interfaces with the database of all these different neurons that we repaired and cloned and functionalized. We enter the recipe. We define the cortical column and the positions of all the different neurons, the first positions of the different neurons, and then we load them. And what you're seeing here, and this is now a hexagon because we're anticipating coupling these columns to build larger parts of the neocortex. But what you're seeing here is the loading of 10,000 neurons, starting in layer 2, 3, layer 1, layer 2, and the blue are the axons and the red are the dendrites. So, as I said, we're packing together in the space approximately, which is about 300 micrometers in diameter, we're trying to pack and position 10,000 neurons. Now, we use other types of rules, such as mini-columnar rules, to give the first initial position. But one's got to be aware that it doesn't help just putting the neurons there. They're going to have to move and jitter and spin in order to get the exact locations that fit the experimental data that says this is where the synapses are. You can see how much spillover there is that is used to connect onto the next column that is sitting side by side. So the next thing we need to do is, once we've positioned them, we've put the neurons first in position, we now need to connect them. And we do two steps. First, we have to structurally position them according to that statistical information that we derived before, and then we have to functionally connect them. So this is just a zoom up of the dendrites in gold and the axons in blue. We have to run a collision detection for every time that the axons touch the dendrites. And they're touching approximately 100 million times. And we do this on the blue gene, so it's quite a challenge because you have pieces of neurons and axons and dendrites all over 8,000 processors. And it is one of the most demanding challenges, which we're working closely with IBM to work on algorithms to do this faster and faster, because right now we can do just the first iteration, but we need to do 10,000 iterations while we spin and jitter the neurons and check that they are structurally positioned correctly and that the locations are exactly according to the lock and key principles that we've derived before. So once we do that, we can now connect these neurons according to statistics of connectivity, which is our initial process. We're going to use algorithms later. But statistics of connectivity says, we've structurally positioned you, you've touched, but only 10% are now going to be connected. And those 10%, when they connect, they will put multiple synapses on each other. And that's how we solve the problem of having random structural connectivity, but specific functional connectivity. And this is more or less what a single neuron looks like, packed with synapses in many different locations, from the tough dendrites to the basal dendrites and around. We can also zoom in on a single cell and color map the source location of the different neurons. Now, as I mentioned to you, we have this lock and keys. The lock and keys translate essentially into a specific color map for every type of cell. So we can use the color map to check to see if we've managed to capture exactly what we see in the biology. And that allows us to start getting the specific color map for every type of cell. And it is different and it is unique, and it depends exactly on what are the different source cells. So this is how we structurally and functionally start connecting the neurons, but now we have to turn these connections into functional connections. They have to be able to communicate electrically. So over the past 10 years, what we've also done is systematically record from several neurons, excite one neuron, which is the action potentials evoked here, and record. Everybody knows it's an electrochemical transduction process. You produce a little analog response in the target cell, and what you will notice here is that from this cell onto this cell, you have this rapidly shut down in the transmission. In a simple sense, this can be seen as a low-pass-like filter, but it's really a nonlinear dynamic synapse. You can also have synaptic transmission onto a different kind of cell where you have rapidly facilitating synapses, and it can be seen as a high-pass-like synapse. Now, it turns out, so every neuron contacting other type of cell has a unique nonlinear synaptic property. So we had to derive for all the major pathways the exact types of dynamics that we're going to use to model the transmission accurately between the different types of cells. So there is a map, and there are specific rules, and we've not been able to record from every one of the several thousand pathways, but we've derived rules that allows us to generalize to those that have not been recorded. You also have maps for the inhibitory inputs, and these allow us now to systematically model the different connections between the different neurons in a biologically accurate manner. This is just more or less what a mapping looks like. You have different classes of synapses, and they're assigned between different types of cells. There's a lot of other parameters, biophysical parameters and physical parameters, that are needed to capture the model so that a neuron, when it fires, will inject the right amount of current at the right time into the target cell, and those have been derived. And we can now go to the circuit builder. Once we've connected it and we've assigned who is going to have synapses with it, we can now assign all the synaptic properties which get inserted into an algorithm that will act as a synapse. And this just shows that we can actually, through the model, capture all the different types of dynamics impinging on the cell. All of these cases, about 30 hertz stimulation of the source cell, and you can see that you have the whole spectrum of different types of dynamics that is synaptic injection into this type of cell. So we can now capture the detailed synaptic dynamics between all the major types of cells in the neocortex. There are many other things. We've also looked at learning algorithms, and this is the spike timing dependent algorithm, which we showed many years ago. In addition, there are many other algorithms, because it turns out that you have to, if you have 20 million nonlinear synapses, you need learning algorithms that are going to align them and adjust the filter functions. And we've done a lot of experiments deriving some of these learning algorithms, and they will be implemented. So at this point in time, we do believe that we have the starting building blocks to a sufficient level of biological detail to begin. It's just to begin. It's to take the first step in building the circuit, simulating it, and eventually visualizing it. So, of course, what we do is we take each neuron. You need a lot of neurons. We are simulating for the first time thousands of neurons, that each neuron has the full morphological and electrical complexity. And so the simplest way is just to put one neuron in each processor, turn a processor into a neuron, and use the MPI messaging as axons. And that's essentially what we do, and we've written 2 software packages that allow us to do this kind of simulations. But the problem is that it produces about 1 terabyte of data in 1 second of simulation. So we needed another supercomputer, because we want to decide which terabyte we should keep for analysis. We don't want to spend 1 second of simulation and then spend 3 weeks or a year or half a year to analyze it, and then decide this is nonsense and start another simulation. So we want to use a visualization to immediately allow us to assess whether this is something that is interesting. We want to store it, we want to save it, analyze it further. And so we went to SGI because we needed a very large amount of shared memory. And they were very nice in that they gave us a 90% discount on this little supercomputer. And this allowed us now to build a small media center in Lausanne, where we can create 3D representations with glasses, and we can sit inside this neocortical column. So it's more like, like feeling like a child. You want to build this puzzle and you want to sit inside and actually see how the circuit works. I'm not sure if it's for science or for pleasure, but it is a lot of fun. So this is a first simulation. Now bear in mind, this is very crude. It's a first simulation of 10,000 neurons, and we're only visualizing 10% of them. And the different colors are the different neurons as they fire. And the visualization was really done in the last 2 weeks. So this is really the very first version of the visualization, and it's going to get 1,000 times better than this. But what this just gives you a feeling of the immense complexity of what you could, in principle, see in terms of having the different parts firing. If you watch carefully now, you'll even see action potential propagation. You see that cell propagates. Another one is going to, the voltages propagate. So you really can now zoom in onto single cells and watch the electrical activity emerging. We can screen and we can select and say we only want to see a certain type of cell, or we want to see a certain ion channel, or we want to wipe out a certain type of neuron and see what's the effect on the circuit. There are 1,001 in silico imaging experiments that can now be done in a very rapid, with very rapid timescale, and that can generate hypotheses for studying the circuit further. So I'll wait for this just to give you a quick view of what it looks like to dive inside. Now we're looking at the top of layer 1, and we're going to go inside while the circuit is active. And if you can see at the top some of the voltages changing in these branches. And this, of course, is one thing to realize is that this circuit is still not calibrated. This is what we call version 2. We're aiming at, by the end of the year, only reaching version 10. So this is not calibrated, but it is the first functional 10,000 neurons with activity, electrical activity, and so on. This is just a single cell. You can extract this one cell out of the 10,000 and decide you want to study just what is happening to one cell. And here you can now zoom in and start slowing down. And if you see interesting things happening, and you could relate that to, for example, synapses changing and so on. Okay. So this is just an example of what we call a biological. Everybody says it's impossible. Why do you do it? There's so many things you don't know. Well, our philosophy is, if you don't know it, don't let it stop you. Okay? So you begin. If you don't know it, you assume it, and you find ways to do experiments to fill in those gaps. So we have the first version, and we're already on version 2, which is a different level of refinement of the biological data. And we're going through progressive steps until we will reach version 10, which will capture as accurately as we can the data that we've collected over the past 10 years, and that should capture a tremendous amount of data that is known in the literature for the past 100 years. There are other things that are needed to be done. You can build the circuit, but how do you calibrate it? So we can calibrate and check each of the parameters, but we also need to calibrate its function. We don't want to put this into a behaving brain, because you can then calibrate anything you want. We want to calibrate that the circuit itself functions correctly. And what we start doing is we do experiments of where we bounce information between neurons. So we record from 3 cells, we stimulate it, it activates another cell and produces another cell. And these disynaptic pathways allow us to start working out properties that if we do the same thing in the simulation, we should see similar statistics of firing other cells, how they fire them, the amount of current that is injected into another cell via another cell. But we can do that now with a 12-patch to an even more sophisticated level, allowing us to look at the circuit and to look at circuit statistics to see that we get the same. These are these little disynaptic pathways that are activated through a second cell. These are direct connections. but we can also do a whole host of dose-response curves where we activate a set of neurons and look at one neuron and see how it behaves. And we can act different sets. So we can get as many constraining principles as we want to calibrate the circuit. Okay, so this is just to give you a feeling. This is not activity, because it took about 5,000 hours to render the activity. But this is without activity of a circuit with 10,000 neurons. And the reason why I'm showing you this is because when I first saw this, I just wondered, what would be your reaction if you look and you see that? Well, my reaction was, what if we painted perceptions onto the dendrites? Forget about the neurons. Let's paint the perceptions directly onto the dendrites. It's analog. So if you were the maker of the brain, would you choose this to map your perceptions and build your perceptions? Or would you choose this? Would you map analog perceptions on there? In fact, it's known that the voltage-sensitive dye maps, all the maps that have been obtained, even fMRI, MEG, most of those maps are not reflecting action potentials. They're reflecting synaptic activity. They're reflecting dendritic voltages. So we've seen dendritic maps, but why do we only look at the zeros and ones coming out of cells? Maybe you can actually produce specific structures on there. So I'm sure you all know that the current paradigm that we're in is called the action potential paradigm. And it's the action potential paradigm because what it assumes is that what dendrites are doing, all those branches, is that dendrites are processing information so that they will pass whatever is the right thing, the final computed information, to the cell body, and the cell body will now send that to the next one to produce this chain of spikes. And across many, many cells, the spatiotemporal pattern of spikes represents perceptions, whether they're subconscious, conscious. It's the spatiotemporal pattern. You can analyze in any way you want. You can do higher order statistics, anything you want. The pattern of spikes are seen to represent our perceptions, and that's what we measure. That's what everybody does in the experiments. They measure the spikes, and they try and see how they can recreate perceptions. Well, what if we looked at the opposite paradigm? Completely the opposite. And we said, and we make a Copernican-like revolution, and we say, let's forget about this. Let's move from being somacentric to dendrocentric, and say that the information is directly represented in the dendrites in analog form, not in digital form. And let's say the perceptions are formed directly on the dendrites, and that the spatiotemporal pattern of spikes just maintain and animate perceptions. They're just the animators. Just what if? This is just an alternative way of looking at how we may interpret the brain. And let's push this a little bit and see where it takes us. So we know that we can get patterns. We can get maps onto the dendrites. And they go across dendrites. You have to realize that for these, right here and right here, it may be the same neurons, two dendrites, having two different complete properties. Or here, it's a collection of neurons from many different locations. So you are getting dendritic maps across the dendrites. But what happens if you could put intelligence into the circuitry so that you could start painting, literally painting pictures onto the dendrites? This is Ramónica Cajal. He's the guy who we had this conference about, who won the Nobel Prize for drawing all these cells. But what happens if we could put enough intelligence in the circuit so that each part of these compartments could be controlled in such a way that you could literally paint 3D pictures onto the dendrites? So it's not that simple because they've only done up to now 2D imaging. But if you go to Amiram Grunwald, who's done most of those, he has in his imagination done 3D, and his imagination has painted these pictures that you have the map, and if you look deeper, there are columns, much in line with Hubel and Wiesel's discovery that you have columns. But more and more, the recordings are suggesting that it doesn't look like that. It more likely looks something like this if you had to do these maps. Now, you have to consider that when you're doing this, one neuron sitting inside this block has dendrites lying all over these properties. How do you get those very specific control of voltages in compartments? So maybe if we did a realistic image, for some reason most neuroscientists just only show bars and stripes and all kinds of things. Maybe if you showed a realistic image, you'd start seeing objects forming inside the dendritic field. And maybe what we really need is topology to help us understand the mathematics of how we build objects for representation. And maybe if you really get it very precise and you can put a lot of intelligence into these circuits, you can start building little people. You start building scenes. And then what you need is you just need spikes to animate these little objects. And it could be that they even become real complex scenes, analog scenes across the dendritic space. So we did a very quick simulation just to play with this idea. We took the dendritic column and voxelized it and threw away the neurons. Okay? And take each of the voltages, and we mapped the voltages collectively within a voltage into each of these voxels. All right? So we're just looking at the brain now as a block of tissue. You've got to remember that 90% of your brain is just dendrites. So we're looking at it as a block of dendritic tissue. This is what happens if you disconnect it. There's no intelligence. You disconnect the circuit, and you just do Poisson stimulation. Nothing interesting. It follows exactly what's the pattern of Poisson stimulation and which neurons you stimulate. But now you just connect it. It's not connected intelligently, but you just connect the circuit. And what you immediately start seeing is the formation of complex structures or objects. Something that deviates from just a simple structure. So here it's just showing you, let me go to another one. That's just a single voltage where EPSPs are occurring across a single voltage range. But this is when you have two different voltages, so you can actually see that the complexity of these objects could actually take on several different forms. And you can actually see there that they do cluster. So somehow the voltages across dendrites coming from different neurons, even when you just do Poisson stimulation, if you have a circuit with some form of connectivity, they start clustering. So what happens if you could really put intelligence into those circuits? This is just to give you a feeling that actually if you zoom inside there, it's not just that it's an object, it's not just that it's a chair, it's an entire universe. You could actually have a whole scene of inside a region of dendrites. So we did experiments, and actually this is totally coincidentally in parallel in another part of my lab for other reasons where we're studying plasticity. We did an experiment to see whether it's possible to rewire the circuitry to insert intelligence into the circuit. So we patch 6 cells, and we see how they connected. So we can define the circuit. Now we take the pipettes out, and we wait 12 hours, and we repatch it. And what we found is that the circuit was different. Not only after 12 hours, but actually after 4 hours. And just to show you how much inertia there is in the current scientific paradigm, Nature said that this was not interesting. Science, I sent it to Science. They said, not interesting. So this will come out in PNAS in another 2 months. So we do these recordings, and we puff glutamate now. Not just, we actually activate the circuit. We can't still put intelligent stimulus, but we activate the circuit. And when you activate the circuit, here you can see that you have connections connections appearing, and you have here connections disappearing. This is potentially the substrate that Jerry Edelman could use in all kinds of restructuring of the circuitry. This is happening, we've done this now, over a 4-hour period. You can still see the circuitry is dynamically rewiring. For 50 years, we've studied only how synapses are getting stronger and weaker, not how the circuit restructures itself. So it is possible through experience, through stimulating the circuit in a particular way, that you could potentially insert intelligence into circuits to get them to build more specific objects. Now, it's also coincidental, and it's just another 2 seconds, that electron microscopy has recently shown that the distance between synapses is so close that spillover from one input is almost impossible to avoid onto another one. Within 200 nanometers, there's another synapse that is related to a different neuron, which means that it may just be that what the brain is trying to do is to control the voltage within a certain voxel compartment, and not so much just that particular neuron. So if we summarize, let's look at a world as an electromagnetic dendritic object, a virtual environment. And what it would say is we do not see the world. We do not see the world. What we do is we use any clue that our senses can provide us to build a virtual analog model of the world on the dendrites. So the world that we see is not the world. The world that we see is the world we build with as much information as we can to get as accurate as we can about it so that we don't make mistakes and bump into something. So neurons learn to contribute to the circuit to build a distributed dendritic object. The circuitry provides the rules to build and animate dendritic objects. Enhanced cognition is the ability to run a simulation of the model into the future to minimize or optimize adaptation. Different brains can learn to build the same model. We are 6 billion people on this planet. Every one of our brains are different. But we use similar rules to build the same world so that we can see the same world and interact. But animals have different brains, and more than likely, they build a different world. They do not build the same world, even though it's the same world, it's presumably the same. We use spikes to transfer the minimum information required to change or transfer perceptions. It seems to me crazy to transfer entire perceptions from one place to another, one part of the brain to another. You just want to transfer those small updates. And spikes could do that. They are rich enough, but not that rich, that they could probably transfer complete perceptions. Spikes emitted on top of dendritic perceptual waves. So you could almost imagine that if you have dendritic waves, that it's just spikes are perhaps the byproduct that are driving and animating our perception. Minimize the number of spikes used to learn to transfer just the required information. It's known for several years that the key thing that happens in learning is that you actually start spiking less and less and less, because you only want to get more and more efficient to transfer some specific information. This also has a radical implication, which is that perception may be there without a spike. And that you may even have perceptions or resting potential contributing to perception. So the challenge to robotics and to people that model the brain and use spikes is this theory just says, which is the contrast to the current paradigm, it says that if you take your robot and you extracted those spikes, you may be able to get enough information for that robot to interact very intelligently. But you will not be able to use those spikes and recreate what we see here. You will not recreate the analog perception that we have when we interact with that world. You will maybe be able to get it to look as if it can interact, but you're not going to be able to use the spikes. So there's not enough information in the spikes to recreate such a complex analog world with every little bit of detail. It's a challenge. I'll just let you read that and just skip straight to the end and say thanks very much to lots of people. Of course, Phil Goodman, who's here, has been instrumental in many aspects of this project. Idan Segev is helping in the single neuron modeling. Charles Peck and James from IBM have been involved in lots of aspects of getting the simulation to work. IBM, of course, just a very big thanks to them because they gave us a extremely expensive machine for very little. And a lot of this work has actually been supported by the ONR while I was in Israel for 6 years. Tom McKenna was the program officer. And of course, Switzerland, thanks very much for agreeing to support with millions a project that doesn't have a clear goal. Thank you very much for your attention.
Thank you very much for this wonderful talk. We have a couple of questions. Please wait for the microphones. Do we have...
They're so hands.
Let's start at the...
Many studies have shown recently that inhibitory neurons in the neocortex are connected, in addition to the GABAergic chemical connections, by gap junctions or electrical connections in dendritic level. I wonder if you have information on those type of connections and whether you have incorporated them into your circuits.
We haven't incorporated them yet into the circuit. There's a lot known, and there are a lot of other people that work on gap junctions, and there's a lot known about how they could be connected, and whatever there's known, we will incorporate into the circuit. It's not an obstacle to incorporating into the circuit. I think one thing one has to be careful about gap junctions is that they are not there all the time. Okay? If you have two neurons that have invested a huge amount of biological intelligence to make them different, if you gap them, you turn them into the same neuron. So my view of this is that gap junctions are important, but as potential moments when you want to synchronize and lose information. But there are things that we can incorporate.
Henry, as someone who simulated 100 compartment model neurons, I recognize and admire the work entailed in simulating 10,000, and I applaud you for that. I want to take issue with one core thing that you touched on about a third of the way through your talk. You briefly outlined the lock and key rules for connectivity as a way to overcome this problem that you encounter when you build a circuit, which is how precisely to connect it. And as I understood it, and I won't try to do it justice, there's statistics of connectivity as a function of branch order between neurons of specific types.
Yes.
My objection there is that still provides you, even though you've got a branch specificity, that still provides order specificity, that still provides you a fairly random result. Maybe that's the tabula rasa that you talk about. Whereas one envisions these circuits growing with activity-dependent wiring, wherein, for example, two-layer force cells that together correspond to a similar feature may send their axons to the same branch, not just the same order, but the same branch in a layer 2-3 pyramid. And so my concern is that the wiring scheme that you described is perhaps a step in the right direction, but without the specificity based on some sort of sensory input, you don't really wire it as you need to.
I understand. That is the reason why I agree with you. And right now we use probabilities of connectivity to decide, and then we just get a circuit. And that's what we can do right now. But what we're refining, we've gone and looked at the molecular causes of this rewiring. And we're turning that into an algorithm, which means that you first start off with a circuit that's unconnected, and you show it a stimulus set, which is the world that it will see, and it will begin to rewire. It wires itself according to it. So in the end, we're not going to use probabilities of connectivity to wire, and you will have very specific wiring. If the dendritic object theory is something that one can explore further, one thing it would demand is that each neuron has to have, every synapse has to have a very specific reason for being there, because it's going to contribute in a very specific way to the object that is formed. And so that is one of the things that we will do, is we put as much of the algorithmic intelligence into it as possible.
Henry, thank you for showing all of us Cajal's butterflies of the soul. but I have a conjecture, and I'd like to try that out on you, then a question. My conjecture is based on your vocabulary. You say a circuit. It seems to me there are multiple circuits, and the question arises how this degeneracy works and how, in fact, you can select one over the other in the absence of input. So I believe that you've already hinted at the fact that there's a multiplicity of circuits. So there's a contingent question which says, if I made another column right next door,
Yes.
would it look the same? That's the conjecture part. Now the question is, what are you going to pick for input?
Okay, so the first part is, it's a little bit related to the previous question, in that I agree with you, there is an issue of overriding algorithm that may start controlling how exactly you get a specific instantiation of a circuit. And what we've seen is that there are such powerful genetic rules. You know, there's this whole debate about nature versus nurture, but in my view, it's a nonsense debate, because 99% of the time, what we see is genetic. And the small amount of changes, we're sitting on top of how a human sees. We don't see like cats see, we don't see like monkeys see. We see like a human sees. And so there is a certain amount of genetic control as to what kind of circuit's going to work. My prediction is for the future that the difference between species is synaptic precision. That the synapses become much more precisely located. And we're going to have to understand how do we get more and more precise synaptic locations. The second question was, what was the second question? Oh, the input. At this point in time, what we're interested in doing is seeing that we have a calibrated circuit. And that requires different kinds of inputs, the kinds of inputs that we can do in experiments. Because that we can reproduce, we can stimulate 1 cell, 12 cells, 1,000 cells in layer 4 and look at the responses in other areas. We want to analyze the circuit and calibrate it first as a chip. Once we're confident with that, we're going to begin to put it together and start to put in the thalamus and move towards whole brain stimulation and have realistic types of inputs.
2 more questions.
This is the first step.
Hi. With the focus on using of supercomputers, I was wondering if you think that the limitations of supercomputers, even as powerful as Blue Gene, will that in any way affect what you're doing in the progress? And do you think that we'll have to use new machines once you get to a certain level?
I don't know if I should say this, but I'm trying to convince the Swiss government to buy a petaflop Blue Gene. They are hesitating, but they haven't said no. It's a very expensive machine, even with a massive discount from IBM. So we can get bigger, but even a petaflop machine is not going to simulate 100 billion neurons. So there are discussions also with IBM in terms of looking at building both FPGA-based computers, specialized ASICs, like the cell processor is a specialized ASIC that is used to solve to solve equations for real-time rendering. But you could, in principle, build ASICs that just solve neural equations. And this could speed up computing by a huge amount. And if you built a specialized computer that just does that, I think that within current technology, without quantum computing and all kinds of other stuff, with current technology, with enough money, you could build a computer that could simulate billions of neurons. I think it's possible.
Different technology rather than having to...
Yes, I think even the current Blue Gene, even if you got the petaflop machine, you're not going to simulate with this level of detail a full-scale human brain. You'd be able to probably simulate a mouse brain, which is still a good step.
As someone who is a theoretician and hasn't simulated or experimented with a single neuron, let alone I applaud anybody who can model well one of them, let alone 10,000. But I do have some questions here, because my naive understanding of how the connectivity works is that, say, a cortical neuron has 10,000 immediate afferent neighbors, and maybe half of those are within the same region, say V1, and the other half are coming bottom up or top down from outside regions. Then if you're only simulating 10% of them in a certain, maybe in certain columns deeper in the cortex, this doesn't apply quite as much. But if you're doing only 10% of them and the connectivity is nowhere near 10% within there, then you're actually the real neurons wouldn't be getting most of their inputs from the neighbors that you're getting.
The secret of the neocortex is that the recurrent circuit within the local circuit is almost 80%. So 20 to 30% of the input is really extrinsic. These are very powerful local recurrent circuits, extremely powerful, and they run on their own. So it's not true there's only 10%. They are very strongly recurrent circuits. The second thing is we know more or less where they're coming in. And those inputs can be supplemented with background activity, with pattern correlations, and so on. So it's not an obstacle. What we refer to as a minimal circuit is that a neuron needs about, most of these cells have about 1,000 to 2,000 other neurons. And so that's why a 10,000 neuron circuit is the minimum circuit.
Well, perceptions on the dendrites is an interesting concept, of course. And one of the things I like about it is that it seems that there's strong evidence in experiments for timing codes, and the timing information seems to be preserved at the synapses, at the dendrites, but has a heck of a hard time penetrating into the soma level and beyond. So there's something in support of that. I have one last question before I hand this off. How did you model the thresholding behavior of the neurons to decide when they were going to spike in response to the stimulation that they got from their neighbors? Was that just a refractory period when they couldn't spike at all, and then a constant threshold, or something more sophisticated than that?
The neurons are modeled with ion channels, where every ion channel is what's called a Hodgkin-Huxley channel. It's still a simple version. We're going to actually go to a much more advanced version of a channel, but currently that's the most popular way of modeling an ion channel. We put ion channels in, and the ion channels, depending on the kinetics you give them, give rise to a nonlinear behavior, which is the actual potential.
I wish to ask regarding the information processing. So you said that every second you get a terabyte of information, and you said that you didn't want to keep all this information, so instead of that, you got some visualization program going on. So I still didn't understand what you decided to keep out of this information, or are you now just
Right now we're not
watching?
keeping anything because it's just the beginning. It's the first version of the circuit.
So you're just looking at the dynamic now.
There's a lot of things that you can look at in terms of why and when different neurons are firing. We've been studying them for 15 years, and a lot of it is just using it as visual diagnostics to immediately see whether things are wrong. We could look at the circuit and see that the certain RI, which is axial resistance, is incorrect because the propagation is too far, and things like that. You could actually use it for a huge amount of diagnostics way before you go to fine-level quantitative analysis.
So the current analysis is by eyes?
Sorry?
So the current analysis of the data is by eyes, by viewing what we see?
We are now at the phase where we're moving into a very large analysis program, and this is where we have a very strong team with IBM and going into analysis in all different forms. And so visualization is a way of pre-screening the data and having fun at the same time.
I know it would be perfect to have another half an hour of discussion, questioning, but I know that we have to move on. Thanks very much.
Thank you. Thank you. Thanks. Thank you.
So thank you very much. We shall now proceed to lunch in the cafeteria.
To meet you.
There'll be people outside providing directions.
Fantastic.
You know my friend Alessandro Vito?
Thank you. Yes, I do.
He worked with